Thanks very much, first of all, to the organisers for inviting me.
Viviane, can I introduce you and say Dr. Viviane Quirke from Oxford, come up. Thank you very much to talk on modern pharmaceuticals. You do the rest.
And thank you for inviting me. I'm very pleased. I'm not a chemist by training, so don't expect me to go into great detail about the chemistry. But I do find that chemistry, the history of chemistry, is a wonderful way, which is too often neglected by my fellow historians. It's a wonderful way of understanding this country and other countries' history in much more general terms. So it's, for me, it's a kind of prism through which I can uncover aspects of history which are often neglected by more mainstream historians. Now, I've spent a lot of time working on the history of ICI's pharmaceutical division, and I'm currently working on a book which will be on the history of its R&D. Obviously, I can't give you the whole synopsis of the book, that would take far too long, but I was very glad to be invited today because it gave me an opportunity to discuss something I've started feeling very much since I've been working on this book, and that is that although ICI was very much a global company, and that's obvious from its name Imperial, it is also very much a local company, which often in a very human way, built on local networks in business, in science, but also personal networks that were forged by people who worked in ICI. And I hope to give you some examples of this, a couple of examples, in what will follow. So, it was full 9, 27 and 26. You see this date is contradicted in my next slide. But anyway, it certainly was formed in 1926. And at the time when it was formed, just picking up where Peter left off, it was Britain's largest exporter of alkali and one of the largest industrial enterprises of its day. As I said, a company veritably with imperial horizons. In fact, I was interested to see that ICI had to fight to have the term imperial accepted in its name. The registrar who registered company names opposed it violently, but I think both Brunham and Mond defended it, arguing that what ICI was going to do would be essential to the economic prosperity of Britain and also the security of its empire. So it was positioning itself already as a global company. However, and this is, I suppose, a major sort of thesis of my paper, it maintained very distinct roots in the Northwest, and it managed to build upon these roots and local, regional connections to succeed in an offshoot of its original business in dyestuffs, and that's pharmaceuticals, which I'll be talking about today. So my paper, after giving a kind of overview of the history of ICI and how it diversified into pharmaceuticals, will explore these local connections, both within the company, but also outside the company, focusing more especially on one pharmaceutical R&D project, the anesthetic halothane, also known by its brand name Fluothane, and I saw that in your museum you have an exhibit on halothane. I'll be mentioning some aspects of this which sort of certainly fit in with that exhibit, but add an extra dimension or a slightly different dimension to it. So let's again go back to the formation of ICI Pharmaceuticals. It started life as the dyestuffs medicinal chemicals section. So a few words about the dyestuffs section. It was the result of British Dyestuffs Corporation merging with 3 other companies to form ICI in 1926. However, and this is quite interesting from a sort of almost social anthropological point of view, it was a poor relative compared to the others, and this had a knock-on effect on its sort of, let's say, its social composition. It tended to attract not public school boys from Oxford or Cambridge, unlike some of the other divisions. I'm thinking of Nobel in particular. It attracted grammar school boys, often very bright scholarship boys who might have gone to Oxford and Cambridge on scholarships, but often went to red brick universities, and often in the north of the country. And this situation was inherited by dyestuffs, and I've interviewed people who worked in dyestuffs, not in dyestuffs, in pharmaceuticals, and who told me that one of the symptoms of this social difference between dyestuffs and other divisions is that in dyestuffs, and later on in pharmaceuticals, people refer to each other as Mr. And they didn't call each other by their first name, which was a mark of being slightly superior, obviously, in the other divisions. So they were very capable chemists who came from the north, from Scotland, from Wales as well, and the jobs that they could get here were an opportunity for many of the brightest and the best from that part of the country. By the late 1930s, however, the situation was reversed, not in these social terms I've just described, but in terms of relative importance and access to funding and general economic prosperity within the group, within the ICI group. And this was thanks to some of the acts that were put through to protect dyestuffs in the aftermath of the First World War, the Dyestuffs Import Regulations Act in 1920, and later on a dyestuffs cartel. And this spurred the dyestuffs division to adopt a much more research and speculative research aspect, most notably with the creation of an azo colour section. And one of the azo chemists employed in the dyestuffs, Frank Rose, would come to play a very important part in the history of the pharmaceutical division. Indeed, he was there as an azo colour chemist when the medicinal chemical section was formed in 1936, and this was following the discovery of the red dye Prontosil rubrum, having activity, antibacterial activity. This discovery was made by Gerhard Domack in Bayer, which was part of IG Farben at the time. However, it was found out quite soon by a French group, actually, working at the Pasteur Institute under Ernest Fourneau, who was a pharmaceutical chemist, that the active part of the molecule was in fact the non-colouring part, and therefore it couldn't be patented, couldn't be protected, and this opened the field for competitors to develop analogues that were also very potent antibacterials. And on the back of this, ICI started committing in a fairly small, modest way with this medicinal chemical section to developing pharmaceuticals, although they'd already been making pharmaceuticals in bulk for other companies to sell. So they had chemical experience of the compounds involved, but they weren't used to adopting a research approach to look for new medicines. At first, interestingly, it was only chemists who were employed in that section, and for the drugs to be tested, for the compounds to be tested for antibacterial activity, they had to rely on testing to be done in local medical schools. So we're already starting to see that they're putting in place local connections for testing the activity of drugs outside. However, very quickly, they decide that it's best to keep that testing done in-house, and they start hiring biologists to work as part of what was veritably starting to look like multidisciplinary research teams, which is what we'd be familiar with today. Now, just like in Peter's story, World War I was extremely important, in my story, World War II has a crucial impact on the future of pharmaceuticals. Partly because it's a rerun of the World War I story, with the realization that once again Britain was relying on German imported synthetic drugs, and that with war looming, which by 1938 was obvious, these would need to be replaced. So in 1938, the Association of British Chemical Manufacturers drew up a list of essential drugs imported from Germany. These included barbiturates, hypnotics, sedatives, anesthetics, and synthetic antimalarials. And ICI, being one of the largest chemical companies in the world, let alone Britain, it was the obvious agent for working on all these compounds, in particular picking at all the patents, German patents, to try and copy the drugs. So ICI was put to work on many of these drugs to try and imitate them, to try to develop their own as well. And ICI was included in major Anglo-American collaborative projects, and I've named those that I've certainly identified that they were major participants on. I mean, we're all familiar with the radar and atom bomb projects, which were joint American and British, and ICI certainly worked on those, but it also worked on joint projects to develop new synthetic antimalarials, which would be very important for the Malaysian campaign towards the end of the war, penicillin, which becomes very important once it's first tested and used in North Africa, in the North African campaign, and then obviously it becomes very important after D-Day, but also nitrogen mustards to be used as anti-cancer agents. Now it's interesting that that work was done at ICI. Nitrogen mustards were obviously being re-explored as potential chemical war agents, but it's interesting that the discovery was made, I think it was 1942, in the US by two pharmacologists, Gilman and Goodman, I think. They found that they were helping to shrink tumors in mice, and I found evidence that certainly the ICI's researcher Arthur Walpole, who would later on go to develop tamoxifen, was working on this as a project in World War II. So in World War II, pharmaceuticals was still done within the confines of the Dice division in research labs at Blackley. But thanks to the switch from a more empirical approach to a research approach, it developed some new compounds. Some of these were antibacterials. Sulfadimidine was one of them, or it's also known as sulfamethazine. It also developed a new antimalarial, paludrine. But it also obviously worked on, developed and made drugs that were developed by others. Mepicrene, which was a German synthetic anti-malarial, penicillin, which they didn't invent but they worked on. And I saw also that in your exhibits you've got pictures of the milk bottles in which the penicillin was grown. And I've certainly, because I've worked on the history of penicillin, and certainly it looks as if these milk bottles were used to culture the mold, but later on, ICI did try and develop a deep fermentation method which was much more effective for producing large quantities of penicillin. They very much did that on their own, which was interesting, whereas most other companies licensed the technology from American firms which developed it first. So companies like Merck, for example. That was the case of Glaxo. Glaxo didn't try and develop the process on their own. But then ICI were quite, shall we say, confident, and they thought they could do these things on their own. They were also inhibited by a patents agreement with DuPont, which prevented them from actually buying in technology from other American companies. So there's a mixture of reasons really why they went in and lost a lot of time in the process into this deep fermentation. Last but not least, there was trilene, which was a purified form of trichloroethylene, which is an interesting compound because it was used as an industrial solvent, but during the war it was discovered it was very good at removing grease from wounds and burns. But then it was found to be useful as an anesthetic agent, so it's quite important for the history of ICI's entry into anesthetics. So World War II had a massive impact on ICI's venture into pharmaceuticals. The projects I've just referred to helped to establish pharmaceuticals within the group, which was certainly half-hearted. Anything pharmaceutical, but medical was thought to be not totally scientific. So, so it helped to establish a credibility of pharmaceuticals within the group. By 1942, enough profits were being made for ICI's main board to establish a selling company, ICI Pharmaceuticals. And by 1944, this was translated into a pharmaceutical division. But I think, more importantly, from my point of view, it also helped ICI to build up scientific and technical expertise in particular areas of which would see the legacy for many years to come afterwards. And one of these legacies is fluorethane. Now, the origins of fluorethane goes back even before the Second World War, with the study of fluoroalkanes, which were developed as refrigerants by General Motors and by DuPont. They were studied, however, as pesticides by James Ferguson, who was working at ICI's General Chemicals division. He actually developed, through his observations of brain weevils basically falling asleep when subjected to these compounds, he developed a very sophisticated quantitative theory of narcosis. And that theory would later make it possible to predict quite precisely which types of compounds would be best to test as potential anesthetics. And this helps to explain, I mean, certainly Charles Suckling, who developed fluorethane, adopted that theory and used it to synthesize very small numbers of compounds and have only essentially a handful of them tested to identify the most active of them. So Charles Suckling, I've already mentioned him, he was one of ICI's chemists hired as a youngster in 1942 to make and use fluorinated compounds, but for a totally different project that had nothing to do with anesthetics. It was to separate uranium isotopes from the tube alloys project, or the atom bomb project. He didn't know, by the way, he's written about it later, and he didn't know that that's what he was working on. It was so secret that even people working on it inside the company were not aware that that's what they were working on. Now, he continued working in ICI's General Chemicals Division, and in the 1950s, Ferguson, who by then had become the head of the General Chemicals Division, decided to launch a new project on anesthetics. Strong from their experience with trichloroethylene, they decided to follow this up with a non-volatile inhalation anesthetic, because the use in operation theatres of electrical equipment was growing, and the use of compounds like ether often led to explosions. So the need for a non-volatile inhalation anesthetic was being felt more and more strongly, and Ferguson, with his experience of working on anesthetics with fluoroalkanes, actually set the task of looking for a fluorinated compound to be developed as an anesthetic. That job was given to Suckling because of his experience of working on the tube alloys project, and he made, as I said, only a handful of compounds, gave them, in a rare example of cross-division collaboration, to James Reventos, who was a pharmacologist in ICI's Pharmaceutical Division. And I think the one that James Reventos identified as the most active in the animal models they were using, I think it was the fourth or fifth one, or something like that, very few, a very low number, was halothane. So it was identified in 1953. It was tested in large-scale trials by somebody local, by an anaesthetist at the Manchester Royal Infirmary. And I've often asked myself, you know, why, I mean, that becomes a kind of trend of using local medics, local consultants, local GPs in Manchester. Why use a local man for this work? Well, it was on the doorstep, it was easy to take the compounds, obviously from Blackley and later on from Alderley Park to Manchester. But also when you're dealing with medics, you need to make sure you control the information that goes to the medic and that you receive back. And I think that geographical proximity is actually quite important in a context like that. But you may have other ideas why this might have been the case, but that's my interpretation. And actually, Michael Johnson didn't just design the large-scale trials, but he played a part in a new piece of apparatus that was needed to give the fluorethane to patients. It needed to be measured very exactly because it was potentially toxic, but I've spoken to another ICI researcher and inventor. I don't know if some of you have heard of Ian Glenn. He developed a later anesthetic, ICI anesthetic, propofol. And if ever any of you get a knee operation or anything like that, you'll probably be getting propofol. He actually told me that he thought that apparatus was there also as a cost-saving exercise because fluorethane was very expensive to produce. Having said that, it might have been expensive to produce. It certainly made ICI a lot of money, and again I've been told by several people I've interviewed that sales of fluorethane were really the first drug that helped to put ICI Pharmaceutical Division in the black. So it may not be a coincidence that around the time that it was launched onto the market, ICI's pharmaceutical division separated from the dyestuffs division. Until then, even once the pharmaceutical division had been formed, there was a representative of dyestuffs on the main board, and dyestuffs remained in charge of the pharmaceutical labs and also the manufacturing plant, which wasn't surprising because they were actually located in Blackley still. But the real breakaway comes when ICI's pharmaceutical division, their research labs, move physically from Blackley to Alderley Park in Cheshire. And I like this image because it shows very clearly that it's a geographical move, but it's also a move from a predominantly industrial site to a site that's more like a science park or a research park as we would think of it today, and which would have helped also ICI's pharmaceutical division to gain a kind of status and kudos and credibility with the scientific world, which they were developing connections with as well. It's interesting to me that the theme chosen for this symposium, marking the opening of the new labs, was the evaluation of drug toxicity, because certainly ICI were at the forefront, maybe because of their background in the chemical industry, they were at the forefront of thinking of drug toxicity amongst companies in this country. It was a relatively underdeveloped subdiscipline, if you like, at that time. And a major focus for Alderley Park's research program was also very unusual at the time. It was a focus on chronic diseases, and these chronic diseases would have been worked on in study mostly. Where's the button? This one. Here, which is where the pharmacological labs and pathology labs were. So it's a very interesting outline. You have, of course, admin, which I like to think as the head or not of the research centre. Chemistry is the backbone. It's really what links the pharmacology with their own animal houses to the bacteriology and virology, so the study of infectious diseases, again with their own animal labs. So really the emphasis and the exciting stuff was going on in the pharmacology and pathology, although there was still a lot of work being done in the traditional areas of sulfonamide drugs and to some extent antibiotics, but the chronic disease stuff would have been done here, with quite different types of animal screens being done here, which you had to protect, obviously, from cross-contamination from the animal work with studying infectious diseases here, which helps to explain that sort of layout. One of the projects that I've looked at, because I've been very interested in this aspect of focusing on chronic diseases again, it was quite unusual for the time in the British pharmaceutical companies, certainly, but more generally in the pharmaceutical industry, was the study of atherosclerosis, later thrombosis, under a South African called Jeff Thorpe. And for that study, he needed to identify what the normal level of cholesterol in the blood was in order to select patients for a clinical trial for the drug he was developing, which was called Atherosclerosis, one of the first hypercholesterolemia drugs. And for that, he actually contacted GPs in the Manchester area, who, of course, under the NHS, had at their disposal databases of patients that were huge and were very useful for the sort of population study you needed to carry out in order to identify patients, potential patients for later clinical trials. So this work did involve close cooperation with local doctors, And it sort of says something interesting also about local doctors being quite happy, GPs in particular, to take part in research. So there's an interesting story there about research activities amongst general practitioners, which is not how we tend to think about them these days. The connections, as I said earlier, were not just business connections, they were not just scientific connections, they weren't just connections with medics. They were also very personal connections, which helps to explain the endurance. They were often based on friendships that were forged in the process of working for ICI. And the most recent piece of work I've been doing has been on something that takes us away from ICI, because the researcher, Steve Carter, who worked on cell pathology at ICI and retired from there in 1980, lived near the Cheshire Derbyshire border in a place called Keppelshume, and he was instrumental in creating a museum some of you may have visited at some point. It was called the Micrarium in Buxton. He wanted to take the art, the science and the wonder of microscopy, which obviously were used for his research, to the wider public. And he was able to maintain good enough contacts with ICI colleagues that even after he left, he was given access to organic compounds. He was able to use some of their instruments and also materials for the exhibits. And one of the most fascinating ones was one of organic crystals. I believe this one might be polyethylene glycol, which was shown under a microscope to melt, because he built a kind of contraption with a pipette, actually, just an ordinary, he used to go and collect bits and pieces from the labs. So he used a pipette to divert some air from an air pressure system he had for controlling the microscope. So he diverted some air which was heated by a coil. He made all this himself, and it started heating the center of the crystal, and as it melted you saw this black patch gradually grow in those beautiful colors which were shown thanks to polarized light. They would disappear, and when you stopped the air, the crystal would regrow, and children or adults who came to the museum could press a button that controlled the rate at which the air came and when it stopped, and when the crystal could regrow. So I want to stop my talk here and say that where do we go from there? So far it's quite an encouraging, quite a lovely story, isn't it? Full of optimism. Well, ICI spun off ICI Pharmaceuticals to form Astra in 1989. So ICI exists as Nobel? So Nobel. ICI's pharmaceutical division exists as Astra, which merged with Zeneca to form AstraZeneca. So it still exists out there, but sadly it was relocated to Cambridge. And I know this panicked a lot of people in life sciences in Manchester who felt they were losing a major resource on their doorstep. Now, the buildings in Alderley Park are still there, and I understand that small companies seem to be moving in as we speak, and Manchester is in the process of also trying to build its own sort of biological, biomedical centre on its own doorstep. So it's not the end of the story, but it's certainly, in my view, the end of an era.
That's great. That's also another unfamiliar story brought to light. Nice.
just make a comment about the Alderley Park.
Yes.
I was on the team that was involved with trying to rescue the place afterwards, and yes, you're quite right, it's being turned into a science park, and it's jointly owned by Manchester Science Parks, Cheshire East Council, and someone else, I can't remember now, and it's being jointly funded by those people to encourage small companies to come in there. And it's being funded by the fact of being allowed to produce some houses on part of the land, and that is giving a fund to allow small science companies coming into the park. So the future looks very good, actually.
Yes.
There are quite a few companies that are really.
Sorry, the architect, I found, for Alderley Park, I found the architects, I can't remember their name right now, because I was desperately trying to get copyright to use their images and their plans, because I think they're so brilliant and so interesting.
Not Fairhurst.
Yes, Fairhurst.
Harry Fairhurst.
Yes. That's who they were, and I think they still...
He died.
Did he? And they looked and looked for me for some sort of, because I would have loved to have had also the artist who painted that watercolour and get copyright from him. So, and you know, it's really sad. I've asked all the people who left ICI, do you have any pictures? Can you let me have any pictures? You know, even portraits of people, and they've hardly kept anything.
Fairhurst is still going though.
Yes. Sorry? It's still going?
Yeah.
Yes.
So that'll
I
very
may
nice.
try again, I may have another go. Yes.
Harry specialised in laboratory.
Yeah.
Gentleman next to Bob, yeah.
Yes. I was going to say that there's precedent for what might be happening at Alderley, because the Heath offices next to ICI Runcorn were closed down, the research laboratories there, and people were extremely worried, but a lot of new people have gone in, and a lot of scientific research has been carried out there, and some of the works that have, inventions have then developed further and then moved into bigger premises. So it's not all bad, but I am a bit concerned about, often's been mentioned about the loss of Objective 1 status with some of the money coming back from Europe, and the North, certainly, we talk about, well, George Osborne was talking about the Northern Powerhouse, that's been forgotten, and I think we're going to have to shout a bit louder.
Yeah, and there's obviously a concern also about archives, is that what you... Yes.
Diana, yes.
Yes, thank you. I'll stand up. Professor Wilson Baker, who became Professor of Organic Chemistry at Bristol, was born in Runcorn and was actually Harry Baker's son. Harry was the first general manager of Castor Works. Have you come across any references to Wilson? Because he was deeply involved with Fleming at Oxford and is reputed the first person to give a paper on penicillin after the Second World War. But was he involved with Blackley?
That would be, I know, because I've worked on ICI's penicillin files, I know there was a very good connection between Flory's team and ICI, and ICI paid visits. I've also worked on the pathology, archives of the School of Pathology in Oxford. And the name Wilson Baker is certainly there. Now, I haven't worked on that for a while, so I can't remember in which connection, but certainly the name is familiar. I didn't realise there was a Runcorn.
Oh yes, he was born in Wilson House just across the ICI guest house.
Yeah. But could I take advantage to ask you then to say something about, the risk to archives, because that's something, the picture may be rosy, but the big problem is when you have major relocations like this, what happens to the archives? And I've seen it happen so often that archives have been destroyed, lost.
Well, I'm glad to. Is Paul here at the moment? No.
You're supposed to talk after the break.
Yes.
Yes.
I better explain. I'm Diana Leach. I used to be the Deputy University Librarian at the University of Manchester. I'm the head of the John Rylands Library, so deeply involved in archives. I'm a trustee here as well, and obviously what we've been trying to do is to bring as many as possible of archives of the chemical industry together. So United Alkalis, we have quite a lot. We have some from Blackley. My school friend here, who's sitting here, we've just acquired her father's archives. He was the works manager at the Hush Hush Works, Randall Works. So this is Sue Toller. And so we've been trying to do that. I am at the moment negotiating with an ex-colleague of Rylands because the Blackley archives have been offered to the Rylands Library in Manchester. And there isn't very much chemical industry archives in the Rylands Library. It would have been wonderful if they'd been offered to us. And I think what's happening with the AstraZeneca archives, they're going to take those which are relevant to the research they're still doing with them to Cambridge. It's the Levenstein, what they call the Levenstein archives and the Blackley archives that I think they wish to leave in the northwest of England, and so they should. Where do they go?
But the problem is, how do they define what's the Blackley archives? Because some of the documents I saw under people's names, for example under Frank Rose, he was very much a Blackley man, who then became an important character, you know, for the pharmaceutical division. But he's a product of Blackley, and you can't just chop him in half. So where do you put, you know, his archive?
Exactly. Well, I'm going to say I'm trying to walk a line between it, because we've been trying to collect chemical industry ones here, and we've actually, for those of you who don't know, we've just rescued a lot of stuff from Bronnemont at Winnington, because Tata Chemicals closed non-plus. And so we've rescued what we can, but of course there aren't the great archival rooms to store things in. But you know, if anybody's interested, talk to Paul and myself afterwards. We're delighted to talk to you, because we're trying to build a real reservoir here. I'm working with James Sumner here in the Centre for the History of Science, Technology and Medicine, with the idea of studentships here for people to work on those collections. So there's a lot which potential going on.
Question back there, yes please.
Can I declare an interest? I worked at Alderley Park for 38 years. My father worked at Blackley before me. You may well be aware that my colleague George Hill has recently written a book on the, I think called Alderley Park Discovered, in which he's tried to capture some of the medicinal research, at least, which has gone on there, in addition to a lot of the history of the site, of the Stanley family, which formerly owned the site, and of the natural history, which is his own personal interest. So I obviously regard that as extremely important, that we should try to capture this information. If,
so I'll write that down if that's all right. So George,
I'll talk to you after if you like. So, in fact, just make one further brief point.
Yes, please.
My experience of clinical trials. At the age of 5, I was tramping around the garden in my Wellington boots, as you do. I picked up a garden fork and stuck it down through my foot. I screamed a lot, and I was carted indoors by my father, who produced from the bathroom a glass jar containing a white powder with a handwritten label that said nothing other than 10,040. He applied this to the wound, and it healed perfectly. And my father was in fact the co-inventor, along with Frank Rose, of the antiseptic chlorhexidine, which is incorporated in the Hibitane and similar products. So I think I was part of a very unofficial clinical trial. Where the material came from, I have no idea whether it came from the research labs or the process plant or the process discovery plant. I don't know, but obviously in those days there was a very different attitude towards safety concerning clinical trials.
but I understand Hibitane was an offshoot of the synthetic antimalarial work.
That's right, yes.
Well, that's a marvellous story, especially as you're here to tell it.
Lucky to be here to tell it.
Okay, thank you very much indeed.